Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder why some nights you sleep like a baby and others you toss and turn? Or why your sleep changes as you get older? Well, researchers have been trying to figure out this whole sleep thing for ages. And a new study might give us some answers about how our sleep patterns change over a single night and what that might mean. This study dove deep into something called slow wave activity, or SWA. Think of SWA as a super important marker of how tired you are and how your body is recovering during sleep. Normally, SWA is highest at the beginning of sleep and then gradually decreases as the night goes on. Scientists believe this decrease shows that the body's need for sleep, like a pressure building up, is slowly getting released. It is a big part of what they call process S, the body's internal clock for sleep. Now, here is the tricky part. All these previous studies looked at SWA a little differently. They used different ways to measure a different math to analyze the changes. And some even looked only at the first few hours of sleep, ignoring the rest. This made it really hard to compare findings and understand if the way we measure SWA reliable, especially as people age or in different groups. The researchers here wanted to clear things up. They asked, is there a consistent way to measure how SWA changes throughout a single night of sleep? And how do things like age and sex influence these changes? So how did they tackle this complicated question? They used what they call a multiverse approach, which sounds super sci-fi, right? Basically, it means they tried many different ways of looking at the data to see which approach worked best. And was most reliable, they used powerful statistical tools called mixed effect models, or MEMS. Don't let the fancy name scare you. Think of MEMS as a way to look at data that comes from the same person over time, like our sleep throughout the night. They can account for the fact that your sleep from one moment to the next is connected, and also how each person is different. They looked at SWA in two main ways. One was looking at SWA in all the tiny 30-second sleep chunks, all through the night. The other was by averaging SWA within each sleep cycle. Sleep cycles are the natural cycles of different sleep stages we go through. They looked at the average SWA, the highest SWA, and the total SWA power in each cycle. To make sure their data was solid, they only included people who had between 4 and 6 sleep cycles, and they excluded data that was messed up by artifacts or breathing problems. Then came the big moment. What did they find? Well, this is where it gets really interesting. They tested linear models, which look for a straight line change, and exponential models, which look for a tapering or slowing down change. And guess what? The exponential models, no matter how they sliced the data, just didn't meet the math requirements. They were like a car that can't pass inspection. But the linear models specifically, when looking at SWA changes per sleep cycle, those ones did really well. They followed all the math rules and fit the data nicely. This was true whether they looked at the average SWA, the peak SWA, or the total SWA in each cycle. This was a consistent finding across different groups of people based on how many sleep cycles they had. Now here's the punchline. In these reliable linear per cycle models, they saw a pattern. They found that on average, SWA did decrease throughout the night. This is what we'd expect if sleep pressure is dissipating. But when they started accounting for age and sex, things got complicated when they adjusted for these demographic factors. The significant decrease in SWA, the part that showed sleep pressure going down, often disappeared. It wasn't significant anymore, meaning it was less likely to be a real effect and more likely to be due to chance or other factors. The researchers found that accounting for individual differences, especially in the early part of sleep, and how these differences are linked to age and sex, really changed the picture. So what's the takeaway here? The researchers found that measuring SWA changes linearly across sleep cycles is a reliable way to look at sleep. It meets the statistical requirements and gives consistent results. However, this reliable method didn't guarantee that we could see a clear SWA decay when individual differences and demographics like age and sex were taken into account. This suggests that previous studies might have overestimated the SWA decay, or DHSP, because they didn't fully account for these individual and demographic factors. This study really highlights how important it is to use the right statistical tools when analyzing complex biological data like sleep. The fact that only linear per cycle models worked well suggests that the way we break down sleep matters. And the big finding that demographics can hide or alter the observed decay is a crucial point. It means that our understanding of how sleep pressure dissipates might be more nuanced than we thought, and that age and sex play a bigger role than just influencing the overall amount of SWA. What does this mean for us? It suggests that the simple idea of SWA consistently dropping throughout the night might be an oversimplification, especially when we consider the real-world variations in people's age and sex. The significant decrease in SWA some studies show might be partly due to not accounting for the fact that people naturally start with different levels of CHE. And these starting points are influenced by age and sex. This is super important because SWA is so tied to our idea of sleep homeostasis and how well we sleep. If we're not measuring and analyzing it correctly, we might be missing key factors that influence our sleep quality and recovery. Looking at the methods, they gathered data from over 1,000 adults who spent a night with home sleep monitoring equipment. They specifically focused on adults between 20 and 59 years old, likely to avoid the sleep changes that often come with older age. They used a type of home sleep monitoring called Type 2 PSG, which is pretty standard. After the sleep recordings, a trained expert carefully labeled each 30-second chunk of sleep, marking it as wakefulness, or one of the different sleep stages like light sleep N2 or deep sleep N3. They then focused on the N2 and N3 stages where slow wave activity is most prominent. And they chose specific EEG channels, known for fewer artifacts. And the results were pretty clear. The researchers tested many different models, but only the linear models looking at SWA per sleep cycle actually worked well statistically. They met all the necessary assumptions for the math to be meaningful. The other models, including the exponential ones and the ones looking at every single sleep epoch, ran into problems. These chosen linear per cycle models explained a lot of the variation in SWA, which is a good sign of a strong model. They showed that, on average, SWA did decrease across cycles, but this decrease was only consistently significant when they didn't account for age and sex. This is a really important distinction. The models were reliable in showing a pattern of SWA decay, but that decay wasn't guaranteed to be statistically significant once you factored in age and sex. This suggests that the interplay between individual differences, demographics, and how we measure sleep can really mess with our conclusion. The strengths of this study are pretty clear. By using mixed effect models, they properly accounted for the fact that data from the same person is related. They also did this multiverse approach, trying many different analytical paths. This transparency is fantastic. It allows them to say with more confidence that the linear per cycle model is the most robust way to analyze SWA dynamics from this type of data. The fact that they found the disappearance of the significant SWA decay after adjusting for demographics is a critical finding. It points to a potential overestimation of sleep pressure dissipation in older studies. However, like all studies, there are limitations. They only looked at one night of sleep in people's homes. This means they couldn't control for outside factors as strictly as they could in a lab setting. Also, they focused on one specific brain recording site, C3. SWA can vary across different parts of the brain, so this might not tell the whole story. And while they excluded participants with very short or very long sleep, the exact cutoffs could influence results. They also didn't look at other demographic factors like gender identity beyond binary sex, or things like circadian rhythms, which are known to impact sleep. So, what is the big picture here? This research suggests that our understanding of how sleep pressure wears off throughout the night might need some refinement. While SWA has long been seen as a reliable indicator of this study shows that when we properly account for individual differences and demographic factors like age and sex, the evidence for consistent, significant decay of SWA across a single night is less robust than we might have thought. It highlights that sleep is incredibly personal. This study really emphasizes the need for more sophisticated statistical methods in sleep research. It's not just about collecting more data, but about how we analyze it. The fact that age and sex can mask or alter the observed decay is a really provocative finding. It suggests that future research should focus on these individual differences more closely and explore how they interact with sleep pressure regulation. Perhaps we need to look at longer-term sleep patterns or consider other biological markers alongside SWA. This research is a great example of how scientific understanding evolves. It challenges some long-held assumptions about SWA and sleep homeostasis, and it pushes us to think more critically about how we measure and interpret sleep data. It's a reminder that sleep is complex, and there is still so much to learn. What do you think about these findings? Does this change how you view your own sleep? Have you noticed big differences in your sleep patterns as you've gotten older? Let us know in the comments below. And if you found this breakdown helpful, please give this video a thumbs up, subscribe to RUBIN'S Mind Lab for more science, and hit that notification bell so you don't miss our next dive into the fascinating world of the human mind. See you next time.